is glucose reacting with proteins in the lens of the eye. By reacting with them, changing their three-dimensional structure, which then keeps them from transmitting light in the same way, and so the lens gets clouded. That's part of the reaction. You can, if you look at uncontrolled diabetics, if you take a sample of the protein from their blood serum, you can actually see changes in electrophoretic mobility because what are supposed to be amino groups have reacted with glucose, and you can actually track the number of proteins that are being modified by the high blood glucose concentrations.
Are all cataracts because of that, or is cataracts different?
I think cataracts are different, although that can contribute to cataracts. I don't know. I'll tell you if I get cataracts from all the sugar I eat. Yeah.
I was thinking of the opposite of that. So there's never really going to be a time when you're at that very steep slope in hexokinase, right?
Right. And hexokinase is almost always working near its saturation.
If it ever got that low, wouldn't you just...
You'd pass out long before that, yes. Yeah, you get goofy here, right? This is goofy. This is dead down here. So. Goofy and dead, yeah.
Sorry, did you say glucokinase just removes the, or sorry, you said the GLUT2 kind of very responsive, GLUT2 transporter is very responsive to small changes.
Right.
But the glucokinase, that just removes the glucose from the blood.
Well, again, it's if we go back to our figure. Right, you go back here. Right? So the liver has two systems that are reacting to blood glucose concentration. One is a transporter system here, that when blood glucose is low in that 5 millimolar range, it's not working very much. It's just relying on its GLUT1, which is still present. But as the concentration of blood glucose goes up, right, this moves into its active range, so you start transporting it in faster.
Oh, okay. And
as you're transporting it in faster, then that concentration is increasing, and so the glucokinase is there to convert it to glucose 6-phosphate faster. Okay?
So is the glucokinase like in the, is it bound to the fructose 6-phosphate for a majority of it until like a lot of glucose is coming into the cell?
That's a good question. I think that's the implication, that at rest, when there's lots of fructose 6-phosphate, it's sequestered in the nucleus so that it's not working, right? So the liver cells at rest are doing, their GLUT1 is active, their hexokinase is active, they're just doing what other cells are doing. It's only when you get that influx of glucose that then glucose inhibits that sequestration process. And by inhibiting that sequestration process, then releases the glucokinase to clear that glucose. I mean, it's a pretty cool and pretty intricate process built on some simple principles. It's one of the things I like about this stuff. Okay? Any questions about that? Let's see. I can talk more about glucokinase and stuff, but again, that's only going to keep us behind. Okay. So, let's look at phosphoglucose isomerase. Right, we said that glucose 6-phosphate is in equilibrium with fructose 6-phosphate. So this is a figure from your book. And the enzyme that does this is phosphoglucose isomerase. And it's the first example that we have of an enzyme family that converts aldehyde sugars into the corresponding ketone sugar, right? The aldose to the ketone. So if we talk about the mechanism of one of these, we will basically cover the mechanism of all of them. We will encounter this mechanism here. We will encounter it later in glycolysis in the triose phosphate isomerase reaction. We will encounter it in the pentose phosphate pathway, in the conversion of the ribose phosphates to their corresponding ketone derivatives. At least those three places, probably more, right? So this is a fairly common reaction scheme. Yeah.
So this enzyme can take it both ways?
Yeah, this is a freely reversible enzyme. Okay. So this is the first of our equilibrium reactions in glycolysis, right? We said there are a bunch of equilibrium reactions that are used for both glycolysis and gluconeogenesis. This is one of them. Okay. So yeah, freely reversible, interconverts, aldehyde and ketone. So flow depends on what's happening. If we're doing glycolysis and using fructose 6-phosphate, then the flow goes this way. If we're doing gluconeogenesis and making fructose 6-phosphate, then the flow goes that way. Right? The flow is completely dependent on the relative concentrations and what is being produced and what is being removed at the time. Okay? So, are we good with its place here? So let's do the mechanism so that we can see that. And it's going to illustrate another thing. Right? This is one, yeah. You're getting good. You can tell when I'm going to start drawing on the board. This is where we have to remind ourselves of another phenomenon that we learned in organic chemistry. And we learned it in organic chemistry. It's nice in synthetic organic chemistry, but it really impacts biochemistry. And it is this fact that if you have a hydrogen, right, alpha to a carbonyl, right, on a carbon alpha to a carbonyl, that hydrogen, normally we don't consider a carbon-hydrogen bond as acidic. But in this case, because of the presence of the carbonyl, and I'm going to draw this all in as one thing, you can get resonance of the unshared pair of electrons so that the oxygen carries the unshared pair. And so that makes this position much more acidic than we would expect. Right? And so there's resonance where there's carbanion character here, but the more stable resonance form would be with an anion character on the oxygen. Right? So this kind of reaction is much more important to us in understanding biochemistry than we give it credit for in organic chemistry. If you remember, this is one of those things that we were rushing to get to at the end of organic chemistry. But in biochemistry, biochemistry takes advantage of this carbanion formation adjacent to a carbonyl all the time. And it's something you're going to get tired of me talking about in this class. Right? Because if we draw the resonance form of this. Resonance form of this is this, with the negative charge on the carbon. So this formation of this carbanion is important. Okay? So this is the first enzyme we're going to talk about where we take advantage of how relatively easy it is to create a carbanion adjacent to a carbonyl, and then to let that carbanion resonate with the adjacent carbonyl. Am I standing in your way, Kyle?
So realistically,
okay. So do you see the situation we're making? Question here? conversation we need to have out loud?
So this actual reaction goes from like the top left down, and then
Right.
it's stabilized by
Right.
the one on the right.
There you go,
Okay.
exactly. So this is the actual, if we were doing it, this is the actual product of the first reaction. This is the resonance, right, double-headed arrow. This is the resonance stabilizing that, yes. I was trying to avoid drawing that, and in the end I couldn't avoid it, so I drew it. Okay? Does that make sense? Okay, so let's look at this mechanism. So there's the mechanism small. Oh darn, I don't have the mechanism large, do I? I didn't do it. Okay, so we only have the mechanism small. So. Okay, now, so here is our glucose bound in the active site. So there's an acidic group and a basic group. They don't really account for what happens with this hydrogen ion. So we'll sort of pretend there's something else there. It may get exchanged with the solvent. This is one of those times when I wish they were a little more specific about what these groups are and what their orientations are. So the enzyme, right, so here's our glucose 6-phosphate, right, our glucopyranose form, right, our cyclic form. So in order to do this, we need a free carbonyl. So the first thing we have to do is open up the hemiacetal ring. So the enzyme first catalyzes the ring opening of the hemiacetal. So this acidic group protonates the oxygen so that it can act as a leaving group, right? As it's trying to get away, this carbon needs a source of electrons. The electrons from this oxygen can fall down, making this more acidic. So either this is exchanging with the solvent, or there's some other basic group there accepting that hydrogen ion. They sort of magically do away with that hydrogen ion, which always bothers me about this figure. Okay? And so that opens up. So if we ignore the arrows and just look at this product, we see now we have our free carbonyl on carbon 1, carbon 2, carbon adjacent to the carbonyl with a hydrogen on it, 3, 4, 5, 6. So here's our 5 hydroxyl group that we opened up. So are we all oriented to what is on that slide now? Okay. So we've opened this up. So we need some basic group to deprotonate the position alpha to the carbonyl. So that's what this is showing. We deprotonate this. Instead of showing the carbanion, your book shows the resonance stabilized enolate, right? Which is what I showed here, right? Now, this is an enolate, but there's also a hydroxyl group here, right? So if this were protonated, it would be an ene-diol. So your book calls this the ene-diolate, 8 indicating that it is a deprotonated ene-diol. Okay, so we get this ene-diolate form. Okay, are we happy with the ene-diolate form? See where it came from? Okay, now is where finish the sentence, Joe. Now is where we can take advantage of this chemistry to allow the isomerization. If this group, which deprotonated here, now can reprotonate this oxygen, if it can reprotonate this, right? So if something deprotonates this falls down, this pair of electrons can go over here. This becomes the ketone, this becomes the carbanion, right? They sort of skip that step. So something deprotonates the 2-hydroxyl. It tries to become a ketone, but in doing that gives this carbon now 5 bonds. So this pair of electrons can come here and be a carbanion stabilized by this new carbonyl. And that can then be protonated.